Good morning, everybody. Hope that you guys are having a good start to your day. I hope your Tuesday went well. Um, what I'm gonna do is I'm just gonna kind of jump right in. Um, not a lot of not a lot of stuff that I need to talk about as far as you know, bringing you up to date on things. Um, there's not a lot of news since our Tuesday class, Monday class. How about that? Monday, that's when we had class. Um, so with that in mind, we are talking about biochemistry. And specifically, we're talking about proteins. Now, if you look in the background here, there's lots of yeah, that's a cool looking molecule, cool structure. Neat, awesome, fantastic. Um, but all of those things that you see in the background, we've talked about a little bit. If you remember on Monday, one of the things that we talked about, or kind of what we talked about overall, was our different levels of protein structure. We talked about the primary, secondary, tertiary, and quaternary structure of a protein. And in our discussions of those different levels of protein structure, one of the big things that we talked about and that I was asking about were what were the forces that stabilize those? Could anyone add to the chat what were the types of forces or interactions that stabilize the secondary structure of a protein? The primary structure is stabilized by covalent bonds, bonds in the backbone. Absolutely right. Then, if we go on from secondary structure, there's the tertiary structure, which is where your amino acid R groups are. And when we talked about those, we talked about polar amino acids, non-polar amino acids, and those negative and positive residues. Your positive residues were arginine, histidine, and lysine. And then your negatives were aspartic acid and glutamic acid. So we talked about a couple of different types of interactions that can exist between those different R groups or between those different side chains, I could say. Ultimately, when we look at a structure like this one in the background here, I'm going to highlight this this one right here. I want you to be able to pick out secondary structural elements. There's your beta strands there, your beta sheets. And those are like this whole thing, starting here and ending, where are we? I think right around here. Um, that is a polypeptide. Actually, I think the other end of our polypeptide is kind of tucked back there. Um, and so this is just a really long chain of amino acids that folds in a particular way and makes a protein. Now, what we're going to be getting into today is basically what do proteins do? Proteins are effectively the workers of the body and workers of the cell. They're responsible for chemical reactions that occur as well as provide structures that make up cells. And if you're looking at this chemical reactions, what I want you to think about is, well, amino acid side chains. Well, those are atoms that make up chemicals that, they are going to help make and break bonds in other molecules. So basically all of those different R groups or those different side chains of all the different amino acids contribute to catalyzing chemical reactions. They also contribute to structures or the structure of a cell, the shape of a cell. Now, there are three major functions or types of protein functions that I want you to be familiar with and comfortable with. First is an enzyme. An enzyme is a type of protein that catalyzes a chemical reaction. That word catalyze just means to like basically make it happen, make the reaction go. Kind of an enzyme is a protein that drives a reaction, I'll say. A protein that drives a reaction. Then proteins are also involved in cell signaling and ligand binding. Now, the binding of one ligand to a protein will cause a signal cascade of protein activity as a response. Now, that word ligand, some people will pronounce it ligand, and essentially that a ligand is a small molecule. Typically, it's a small molecule. I think the most common example of a protein that binds a ligand would be HB, not to be mistaken with the movie Hellboy, but instead hemoglobin. Could anyone add to the chat, what does hemoglobin bind to? So hemoglobin's our protein, and it binds the ligand oxygen, or O2. Hemoglobin binds oxygen, and absolutely, Chris, whenever it binds oxygen, whenever I do this, the inhaling, I had to exhale because that's what I do. But the inhalation, bringing all of the air into my lungs, in my lungs, there's little alveoli, and those alveoli have these very small pores that allow red blood cells to bind oxygen so that that oxygen can flow through my bloodstream with the help of hemoglobin. Hemoglobin's the protein in my red blood cells that binds to that oxygen and brings it to my fingers, brings it to my toes, basically perfuses that blood all around my body. And so in this case, hemoglobin is our protein and O2 is the ligand. So that's one part of this. The other part of this is cell signaling. Okay. So what I think is a good example to think about a signal is all of the cells in your body, they have a membrane. And I always kind of think about a membrane as basically like a fence. It's to keep the stuff inside of the cell in and then outside of the stuff out. There are things that come in and there are things that exit. Um, but there's also things that you don't need to get into a cell, but a cell is going to respond to. And so we're going to get into cell signaling and look at a couple of examples. I'll show you a couple of examples of what this looks like whenever we get there. But that's going to initiate a signal cascade, and that's kind of like it is a way in which a something outside of a cell stimulates a response inside of the cell. And then the last major role that a protein plays, or that proteins play, is their structural proteins. They basically hold everything together. So to illustrate these 3 points, we've got on the left-hand side, enzyme catalysis, where we have this molecule right here is our input. That molecule goes to our enzyme right here, or our protein. And our enzyme slash protein, I'm going to use those terms interchangeably, it's going to make a little bit of a change to this molecule. And what I want you to see here is that this little thing that I circled there, and this is what do you notice? I'm going to zoom in. I think that's R4. Can't really tell very well. But when I zoom in, it looks like R2. So in our input, we've got that R2, and then on the right-hand side of our enzyme, we still have that R2 on the top model and then the bottom model. Are those R2s connected in the same way to the molecule as before? Yes or no? So the structure on the left versus the structures on the right, do they look the same? Specifically, kind of looking at that R2 there. They look similar, but there's a very subtle difference. And that subtle difference is, if we zoom in a little bit more, if you look at the R2 right here, that's a little triangle. And then this right here is a triangle, but it's actually kind of a dashed line. Let me go back and I got to zoom out so I can draw it in a little bit. Oh, too far, too far, too far. Okay, there we go. Um, what this is basically showing is we got this, and then this wedge. This is kind of a denotation that chemists use to give a little bit of perspective and depth to a picture. So S and then CoA. So what changed here was we've got one thing right there. We've got a double bond, and now we have single bonds and a single bond. In addition to that, this wedge and then this right here is kind of a dashed line. R2, R2. That's just to show that basically the molecule on the left, something about it changed. The double bond was broken, and then we've got this R2 that's coming out, and then we've also made a copy of the molecule where the R2 is going in. Now, I don't want you to stress about this and be like, yes, exactly, R2D2. I don't want you to think like, oh, do I have to know that structure and that change? No, what I want you to recognize about this is that, well, our enzyme is going to take an input and make a change to it. It's going to do something to change that. And in this case, it took a double bond and made it a single bond. And then it kind of repositioned different atoms. Um, so then our image in the middle, what I was talking about was here is our cell. We've got this kind of fence, and then we have this protein that, well, it takes on this, that kind of looks like a, I don't know, like corn pops or something, or a Kix piece of cereal that is going to bind to this, and then this is going to stimulate some sort of response where it's like, hey, it's kind of echoing a signal, like, hey, there's something outside. I need to let you know. And then what's going to happen down here is something inside the cell is going to change. We're going to again go into more detail on all these. And then last but not least, this structure on the right is an example of one of the critical roles that proteins play, in that they basically hold a shape. They hold a shape to a protein, or they hold a shape to the overall structure. Now, the first thing that I want to get into is enzymes and what they do. So, what does an enzyme do? Well, a living system uses enzymes to speed up and control rates of extremely important biochemical reactions. You could say that enzymes are the agents or the responsible parties of metabolic function. And what I mean by that is, like, are we going to be burning sugars to get energy, or are we going to be burning fat? Do we need to burn fat, or do we need to burn sugar? That's what an enzyme is going to do. One of the many things that enzymes are going to do. And it's not just a matter of, well, do we need to burn fat or sugar? Instead, it's kind of like, should we should we build up sugars so that we can save them in the form of glycogen for long-term storage? Now, an important part or an important structure of an enzyme is known as the active site. And another important term related to a enzyme, and this particular enzyme is one known as lysozyme. Um, with So we've got our enzyme, and what colors do you see that our enzyme is illustrated in? I'm going to say white, and then what other colors indicate our enzyme? Or parts of our enzyme? Okay, we've got red and blue. Okay, now, Stanley, I'm happy, and Caitlin, you guys referenced the black part of this enzyme. Okay, so I'm happy that you saw that. That black part of the enzyme is actually the molecule that the enzyme is going to act on. So that is the thing that is going to be modified by our enzyme. And that is commonly known as a substrate. So I mentioned previously protein ligand binding. Well, there's a really subtle difference between ligand and a substrate. A substrate, well, a reaction, actually, I'm going to change that. A substrate gets modified. A ligand, by comparison, is unchanged by a protein. So I'm going to say a substrate gets modified by, and I'm going to use these terms interchangeably, protein slash enzyme. A ligand is unchanged. So as an example, I mentioned hemoglobin. When I inhale, the hemoglobin proteins within my body, they bind to O2, they traffic it around my body, and then they release O2. That O2 is unchanged. That oxygen is unchanged. So a ligand doesn't get altered, a substrate does. Now, in this model, in this depiction right here, that black molecule in the middle is our substrate. So something's going to happen to that substrate that this enzyme is going to do. Now, the different regions that are white, blue, and red, well, the blue region represents our substrate binding site. So basically, the part of the protein that the substrate is going to be like, aha, that's where I fit. Then the red region is the active site. Now, I highlight these because these two regions, well, certain important amino acids, I'll say IMP, amino acids are found here. And those amino acids are unique because they are able to help make or break a bond with that substrate or on that substrate. Okay. Can I get a thumbs up, a yes, emoticon, whatever, in the chat in response to this, indicating that, okay, I'm good, we can move on. Okay. Awesome. Now, next up, this is kind of removing some of those. Okay, I gotcha. Um, so when we look at this, I want you to remember what a substrate is, that an enzyme has a substrate binding site, and that an enzyme has an active site. So I gave you this model, or I think that I gave you this model the other day. Like this right here is a protein. We'll say this is an enzyme. And let's say that this little headphone that I bought off of Temu, that it's all right, could be better. This is going to be bound, and my enzyme is going to break that thing in half. And if I squeezed hard enough, it probably would break because did I mention I bought it off of Temu? So it's ready to break. Okay. So a ligand and a substrate, those two terms are not interchangeable because a substrate gets broken, gets modified by an enzyme, whereas a ligand, the protein just binds onto it, moves it, releases it, sort of thing. Or it binds to it and just holds onto it. It's like a bank account of sorts. Or yeah. Um, Here what we have is a model for an enzymatic reaction. I'm going to label three things: A, B, and C. Which one of these do you think is the enzyme? Add it to the chat, A, B, or C. Great. Perfect. Okay. Now, A and B, if C is our enzyme, A and B, well, this is building on what we just saw. A and B are examples of substrates. So what that means is that an enzyme doesn't necessarily have just one substrate. An enzyme might have two substrates. An enzyme might have three substrates. Now, to illustrate that point, we're going to walk through kind of the sequence of events that happen. So we have our enzyme, we have substrate A, and another substrate, substrate B. This model of our enzyme How do we know that this design is a substrate and not an active site? Okay, that's a great question, isn't it? Okay, so how do we know that this design is a substrate and not an active site? That's a great question because um, whenever I'm illustrating like an active site, I'm going to do something like this. I'll say the red region here are the amino acids found within the enzyme that make up the active site. Does that make sense, Andrea? If that does not make sense, okay, cool. Is there a max number of substrates that there can be? I'm going to say, I'm going to say no. And with that, I will add that most enzymes, I would say, if there's 25,000 different enzymes known to man or known to people, I would say 24,000 of them have one or two substrates. That means about 500 of them have maybe three substrates or four substrates. Um, so the answer to your question of is there a maximum? No, but is there like an expected maximum? Like how many would I expect to see? Yeah, probably most commonly you're going to see enzymes that have two substrates, one or two substrates really. And I think that whenever we go through this a little bit, that will maybe make a little bit more sense as to the why there. But that's a fantastic question. Both of these are great questions, guys. Um, so what we see from here to here is our substrates both find a binding site. They both interact with our enzyme, and they're both kind of captured by the enzyme. Now, what is the fate of these? So before we get to the fate of them, I want to introduce you to some kind of terminology that's pretty commonly used. So if you remember back in 1310, this is one of my favorite things about chemistry, is you'd write things like shorthand, like cm is centimeters. Okay, so now I don't have to write the whole word centimeter out. That's great. That's a relief. I'm happy about that. Then mole. Oh, I'm gonna write mol. Oh man, that's such a relief. I don't have to write that letter E. Ah, wow. It saves a lot of time. I'm so thankful for that. I mean, it kind of makes sense, but also it's kind of silly. Um, I want to get you guys familiar with some of the terminology that's typically used. And whenever we do that, does anyone want to guess what the letter or the terminology that's commonly used to denote an enzyme is? And I'll give you a little bit of a spoiler. It's E. E is the common term that we use to denote an enzyme. What do you think S stands for? Yep, substrate. Again, these are really complicated, tricky letters to keep in mind, so bear with me. Okay. Now, this combination right here, and I will say it can get a little bit more complicated whenever you're showing a model with with multiple substrates. And you'd substitute, you use different letters. But whenever we talk about an enzyme, the notation that I'm about to show you is really kind of referencing a single enzyme and a single substrate. So what we're going to see form is known as an ES complex. That's our enzyme, substrate. They are bound together. Now, if we jump back and we look at this right here, my question for you guys and add to the chat, do we have the ES complex? Has the ES complex formed in this model right here? It has not. So the way that we would describe this is we would kind of write it down as an equation and we'd say E plus S. That means that our enzyme is free and our substrate is free. And like I said, if we maybe we'd do something like E plus A plus B denoting our enzyme and then our two different substrates. You would add something to like a legend or a key. It's almost like reading a map. You would indicate that. Okay, so here's our ES complex. This is really just showing that our enzyme has bound to our substrates. Now, the next thing that we're going to look at is, well, what's our enzyme doing? Our enzyme is catalyzing a reaction. It's almost like our enzyme is a matchmaker of sorts, in like a dating coach or something like that. I don't know that a dating coach actually exists. They're in movies, though. It's always like a thing. Here's the lonely billionaire who has yet to meet the love of their life, and lo and behold, the matchmaker finds them, but it was the billionaire is in love with the matchmaker the whole time. My father-in-law watches lots of Hallmark movies, so I periodically see those. And that's like the premise of almost all of them. Um, so our enzyme is going to bring these two substrates together and cause a reaction. How does it cause that reaction? Well, the enzyme has this very unique shape. And with that unique shape, substrate A binds here, substrate B binds here. And then what's very important about proteins is that most proteins are your enzymes that catalyze reactions, they move. They have flexibility to them. So if this is our enzyme and it binds, let's go with, if our enzyme binds headphone case and headphone, which is going to work, guys. Yep, there we go. What this enzyme has done is it's brought our two substrates in close proximity and it's positioned them perfectly. And then it makes a minor change and changes its shape. And then what it does is it brings those two substrates so close together that they simply have to react. And lo and behold, look, it put my headphone back in my headphone container. So an enzyme basically puts things in the right place so they can react and undergo some sort of change. It's almost like um, if the enzyme was not there and substrates A and B were just kind of floating around in the universe, would they hit each other? Yeah, potentially. Would a chemical reaction take place? Possibly if they hit each other perfectly, but when the enzyme brings them together, it puts them in the right place next to one another, positioned properly to collide for a reaction to take place. So our enzyme changes, and the result is substrates A and B react. Now, to denote that that reaction is taking place, we adjust our ES, and we use that little asterisk. This is one fun thing about enzymes is what does that asterisk mean? Basically means chemistry. You could also say that asterisk means magic. A reaction's taking place. Great. Awesome. That's magic, not major. Okay, so magic, the reaction takes place. Those two substrates are in close enough proximity, they hit, they can react. Now, let's talk about the aftermath of that. Well, remember we had substrate A and substrate B. Whenever those two substrates come in close proximity to one another, and I'm going to go back, this is substrate A. Bear with me as I jump back. Here's another picture of substrate A. Substrate A, we've got this gray ball, blue, olive colored, brown, and then in the foreground, we've got this one that it's kind of like a purplish. What color would that be? I mentioned this in my last class, but if you've ever seen the movie The Devil Wears Prada, magenta, that's the one I'm thinking of. I always like the right names of colors, and that movie that I mentioned, there's a whole scene where they talk about, well, that's not blue, that's cerulean. And I always like that word. So that's not cerulean. Andrea, you're absolutely right. It's magenta. Okay, so that magenta ball in the foreground there, That magenta ball goes from substrate A, and now it's a part of substrate B. So my question is A the same? Is A here the same as it was on the previous slide? Yes or no? Does it have the same kind of? No, it doesn't. You're absolutely right. It does not. So can we call it A anymore? No. So that's why we have this. A has become C. And if you think about a chemical reaction, there's some terminology that I want you to be familiar with. And actually, if you think back to 1310, what were the things on the left-hand side of the arrow? What does that X correspond to? I'll give you a hint. It starts with an R. Reactants. Perfect. Great job, Chris. Okay, we've got our total reaction here. Our reactants on the left-hand side, what do they become on the right-hand side? Starts with a P. Product or products. Absolutely. Great job, everyone. Okay, so if A was our substrate, that's similar to being a reactant. So then what is C? C is a product. Okay. B is also a reactant. And now B has undergone a change where it's taken on that magenta ball and it is now D, or a product. Part of substrate A has moved on to substrate B. And in the process, we've made two products. Okay. Does everyone follow that? Can I get a thumbs up or a yeah, sounds good, or an emoji emoticon? Okay. Nice. Great job, everyone. Okay, I got it. Okay. Okay. Thanks, Caitlin. I was curious if that was a help, and then I was just like, is this a help I need clarity, or I'm in need of help? Anyway. Okay. Good. Okay, so now my question, I appreciate the enthusiasm. Now my question is our enzyme bound to our products? Or are our products still in contact with, are they still bound to our enzyme? And it's not a trick question. Look at the picture there and, just, yeah, yes, our enzyme is still in contact with and still bound to our products. Okay. So we used E to denote enzyme, S to denote substrate. Just between let's see, we got the 31 of us in here. What single letter do you think that we should use for product? I mean, we've used two good letters, so we're really perfect. We were down to 24 letters, but we got P as our product. So, oh, ESP. Isn't that where you can read minds? Or it's almost ESPN. Um, you're right. P is what we're going to use to denote a single product. In this case, we've got two products that are coming out, but this right here, this model of our enzyme and our products when they are still in contact, this is referenced as and denoted as the EP complex. So we previously had the free E, free S. Then we had the ES complex. Then we had that ES with the star, the asterisk, indicating reaction takes place. And now we've got our enzyme and our product, or our products in this case. Now, after your products are made, you're done. Job's done. So what can happen? Your products are released. Then what else do we have? So, you know, in this case, we're looking at E plus C plus D. free enzyme, free product, free product. Our enzyme, E, it's ready to go again. It's ready to bind the substrate and basically catalyze it again. If you have only one substrate, the convention is to write E plus P, your enzyme plus your product. Now, with that in mind, when you're thinking about this and you're like, what's this going to look like on a test? Um, if I give you a question where I say enzyme E, or I'll say enzyme, I will make it a sentence. An enzyme binds substrate. Let's go with R. Um, what denotes where the reaction is taking place? Okay, if I gave you that, I would give you answer options like this. E plus R, ER, ER asterisk, and then none of the above. I just want to stress that because the convention is to use E, S, and P for your enzyme, substrate, and product. If I give you a question where I redefine or identify what the substrate is, and I say the substrate is R, I'm not going to give you these options and then say, oh, but what about ES star? That would be, I would not do that because, to be blunt, that would be a really dumb, well, maybe it wouldn't be, yeah, it would be dumb. It would be very cruel of me. And I don't believe in that because I think that you'd look at that and you'd be like, well, ES star, we talked about that as the enzyme substrate, but then he also identified R as a substrate here. If you answered ES star, then you would have communicated to me that you understand what the asterisk or the star means, that that's when the reaction's taking place. So in the event that I redefine anything, that would be a part of your answer options. I don't like to have like questions that are just like, yeah, that's cruel, that's brutal. That doesn't really serve anything. Okay, so to sort of sum this up, here's all of the different kind of stages. Your free enzyme and the substrate, or substrates, is E plus S. The plus indicates that they are not in contact with one another. When the substrate or substrates are bound, that's denoted as ES. You use the ES star to denote the reaction is taking place. And then EP would be, well, your enzyme is still in contact with the product. It's almost like the thing has been built, but it hasn't been released to the public. And then last but not least, our free enzyme and our free product. So these are kind of all of the general steps that every enzyme is going to go through. Now, in a couple of days, we're going to talk about the process by which your body utilizes sugar, how it utilizes glucose and extracts energy from that, um, and what are the different players there. And so, you know, you could kind of think about this as an enzyme has to bind a substrate before it interacts or before a reaction is catalyzed. It's not like Harry Potter where it just like alaka-woo, zulu-boo. I don't really know Harry Potter, so I just know that he says things like that. So it's not going to, from a distance, be like, ha ha! No, what is it? Expellius from the Bellius! So it's not going to do that. Instead, it has to like grab the thing and then be like, Expellius! Topicus! Removicus! Oh, uh-oh, uh-oh, uh-oh. Undoicus, computericus. There we go. I fixed it. Absolutely. Okay. So that's like what an enzyme does. So when it comes to this, and you might be thinking, well, what are some questions that could come from this? This right here, to me, is a really good slide to understand. Understand the terminology and how they vary. Well, tell them welcome to class. Um, so this is good, good. Um, these are all important terms and all important like denotations where we learn about or where we can kind of communicate a reaction that's taking place. What stage is it in? Is it in the, yes, the enzyme has bound to the substrate, but the reaction hasn't taken place. Now, one thing that I want to highlight here is this right here. As much as it kind of looks like a smiley face of someone with really intense eyebrows, as much as it kind of looks like that, um, another thing that this denotes is this right here. Okay, so this isn't this notation right here. When you see a single arrow and you think about a chemical reaction, we have our reactants and our products. I should use R. Reactants go to products. When you see an arrow like this, or you might see it written like this, then you got reactants and products. What do you think that means? So let's look at, tell me what you think. Okay, so the one on the left here that I'm going to label left, that means our reactants make our products. Then the one on the right here, what do you think that means? First of all, do you think that it means that our reactants can make our products? Answer that question, yes or no. Can our reactants make our products? Can our reactants be used to make products? Absolutely. Okay. Now, I would agree with that because we've got this arrow that's, well, it's going, our reactants are going towards our products. But then, are our products also going towards our reactants? Answer that. Yeah. So when you see an arrow like that, it kind of forward and backward arrow, you could describe this as reversible. So that means that when we get to this ES complex, so when we go from E plus S to yield or to produce ES, and we see these reversible arrows, that means that, well, that ES complex could fall apart, could break down to our free enzyme and our free substrate. So when you see these arrows, reversible. Now, a great question to ask then, or a great thing to think about then is, well, what is that? Huh? Why? What's that mean? That just means that the enzymes that are in our body and in our cells, they are dynamic. They are responsive. Depending on the environment, they will say, oh, well, I can go backwards. So they can, an enzyme can kind of sense the area and be like, hey, I've been working really hard to make this product, and I can see that there's a ton of this product around. So you know what? I'm not going to catalyze this reaction. So I'm going to release the free enzyme from me, or sorry, I'm going to release the free substrate because there's a ton of this product around. I don't want to add to more of the hassle. Can you add to the chat if that makes sense? Okay. Okay. So my point there is when you see that forward and backward arrow, think, okay, that means it's reversible. Just as we made the product, well, that product could go back to make our reactants. So that E plus S could yield the ES complex, but that ES complex could break down. Okay, so these steps, this is a really important, I mean, this right here, if you've got this, I could probably come up with like 10, 15 questions on an exam just based on this slide. And I don't think that those questions would be like brutally difficult. I think they would be like, what does this mean? What does that mean? Where is this likely to go? And so now that I'm thinking about it, maybe not 15, but I could get between 5 and 10 questions on this, and I think they would be good questions, not just like throwaways. Okay, so that's what an enzyme does. Now, next up in our kind of journey through our different types of proteins, we've got cell signaling and ligand binding. Okay. Now, a substrate is something that an enzyme acts on. A ligand is a small molecule that, well, binds and doesn't change. No change takes place to that. Okay, there we go. So when it comes to, I don't know why I had nothing there, but here we go. When it comes to cell signaling and hormones, hormones are great examples of molecules that are involved in cell signaling and signal transduction, which transduction is just this awesomely complicated word to mean like sending the signal on or relaying, basically. So, EGF here is a hormone, it's a non steroid hormone known as endothelial growth factor. If I remember correctly, HGH, which does anyone know what HGH stands for? I believe that HGH is also a, well, does not enter your cells. So HGH, also known as human growth hormone, kind of behaves in the same way. So for a non-steroid hormone or for steroid hormones, it's pretty common for there to be a, what is going on with, that was a completely useless set of slides. Rats. Ah. Okay, here we go. Endothelial growth factor. I know, I know. It was that, you're right. This, I can only blame myself for that. So I apologize, everyone. Endothelial growth factor. If you hear that term growth factor, or if you think about like human growth hormone, I think it's pretty reasonable to conclude that human growth factor or human growth hormone, yeah, it's probably going to, I don't know, cause something in your body to change. And it changes like an exterior view or the like what you look like. In order for what you look like to change, something internally also probably has to change. So endothelial growth factor, EGF, does not enter the cell. And so think about this as like it is the person who can't get in it, yeah, this is the person who can't get into the bar. Um, but they see one of their friends who is in the bar. This is this is perfect. This is all coming together. Oh my gosh. I can't believe I've ever thought about this before. This is perfect. Okay. So endothelial growth factor is the person who they go to the bar and they're like, "Oh, I gotta tell Jake that after they're done, we're going to Taco Bell." That's my signal. That's what I gotta do. So the non-steroid hormone, endothelial growth factor, is just like its purpose is tell Jake, meet at, no, let's be realistic. People don't want to go to Taco Bell. At Taco Cabana. My kids know Taco Cabana as the purple place. And every time I go to the purple place, um, I'm like, did I just spend $31 on breakfast tacos? Oh my gosh, I did. And then I get cheap and I'm like, next time I'm not going to get cheese. I'm just going to have cheese at home and I'm going to add it to them there. And then I do that. And then my kids are like, you just added the cheese at home. And I'm like, I did. and I saved $6. So your endothelial growth factor can't get into the bar, needs to tell Jake, hey, we're going to Taco Cabana. So what our non-steroid hormone does is it finds, oh, let's see, do I have a, okay. It finds, yeah, here we go. It finds a receptor protein. So this is somebody kind of like, let's say that the bar has an open window or something like that. They're standing where they are physically in the bar, but they're also kind of like hanging outside of the window. Okay. So they are behind the metal gate, but they're inside and they're outside. They're kind of inside and outside simultaneously. And the non-steroid hormone, endothelial growth factor, goes to this receptor protein and says, hey, I'm here. And then the receptor protein says, well, that's great news. And EGF says, hey, I need you to relay a message to Jake that, we're going to Taco Cabana after. Endothelial growth factor, which is the person who is simultaneously inside and outside of the bar, says, I can't see Jake, and I'm stuck right here. But I see Steve. Steve is this other protein. And now this right here is a bunch of alphabet soup. And actually, I'm going to jump ahead. Okay, there we go. So this whole thing right here, all of these are proteins inside of the cell. Okay. Now, for our purposes, endothelial growth factor, here we go. This is what I want. Endothelial growth factor is person who has the message, tell Jake, go to TC, Taco Cabana. What did I say our EGFR is? Or okay, I didn't say a name there. EGFR, endothelial growth factor receptor, that is the person that EGF knows. EGFR, the receptor protein, let's call him, I gotta get names. EGFR is Evan. Yeah, E. Evan tells Dave. Hey, Dave, tell Jake that we're going to talk about Cabana. Dave says, I can't see Jake, but I can see Steve. And then Steve says, okay, cool. Well, I also cannot see Jake, but I'm going to tell Tina that they should relay the message. And then Steve says, hey, Tina, tell Jake we're going to Taco Cabana. Tina says, well, I can't see Jake, but I can see Elizabeth. And we'll see what Elizabeth does. Then Elizabeth says, okay, similarly, can't see Jake. Elizabeth goes and finds Nate. Nate says, well, likewise, can't see Jake, but I can tell Marcus. And, Marcus can relay it to Jake. Marcus says, listen, can't see Jake, but I can tell Beth, and Beth can, Beth knows lots of people. I don't know if Beth can see Jake, but Beth knows lots of people, so Beth will tell everyone and it will get to Jake. And I think I've run out of names that I can come up with. That's right. I've got like 7 names, even though I've got everyone's name sitting right before me. I'm not going to identify any people here. So I'm just going to say Beth tells a handful of people, and then lo and behold, what happens? Jake gets the news. Jake gets the news that Evan is trying to rally a group to go to Taco Cabana so that they can spend $35 on chips and queso and be very happy about that $35 worth of chips and queso. Now, that is the parallel of what happens. There was a message that was outside of the cell that could not get in. EGFR is that message. There are a bunch of proteins involved to relay that message. It's kind of like whenever I know when I was in elementary school, we played that game Telephone, where it's like a line of 30 people and you whisper something in someone's ear, but the message gets all distorted whenever you do that. And that's kind of the game of Telephone, where it's like, yeah, I'm out of ideas. Um, this message stays outside of the cell, or the messenger stays out of the cell because all that it does is it stimulates a protein, a receptor protein. So this R, that's the thing that I want you guys to remember, a receptor protein. That R stands for receptor. Now, the parallel or the analogy was, hey, we're getting taco cabana. What happens in a cell, more than likely, is something like this. New genes are expressed. Alternative to new genes getting expressed, maybe it's going to be activate, and that sounds like it's from a comic book or something like that, or the Avengers. Activate fat burning. Okay, Stanley, great question. Which level do R groups interact in protein structures? Are you asking that in the context of this? Oh, okay. R groups, it to answer that question, it depends on if you're talking about the tertiary or the quaternary structure. The quaternary structure is where there are multiple polypeptides that have to interact, and R groups do mediate interactions there. If there's only a single polypeptide, then it's the tertiary structure. Okay. So, moving on from there, that's signal transduction, signaling basically the way in which signals are communicated within a cell from outside to inside. Now, next up in our last type of proteins, structural proteins. Keratin is an example of a structural protein. There's lots of different proteins in humans, and structural proteins are generally they they're very rigid. Um, an example of a structural protein would be keratin. Um, then collagen is another one. And then let's see, another, okay, keratin, collagen, actin. Does anyone add to the chat if you don't mind? Does anyone take a collagen supplement? I know that my wife has collagen in her coffee. I add it to her coffee when I make her that in the morning. And collagen is a protein that really one of the purposes that it's supposed to serve is that it's supposed to be something that like strengthens your hair and strengthens your fingernails. And yeah, so that's a structural protein. Another example of a structural protein is fibroin. Fibroin, which makes up silk, can be a very, very strong structural protein. So when you think about different proteins and these three different types of proteins, um, structural proteins can be very, very strong um, in the sense that, sorry, let me also add to this. Their sequence. Does anyone remember that what that one followed by the not symbol represents? Add to the chat what that is. Close, close. Primary. First, primary, yeah, pretty much the same thing. So our primary structure, does anyone remember what the primary structure of a protein consists of? It's stabilized by covalent bonds, specifically peptide bonds. The word that I'm looking for, Andrea, perfect. N to C, the amino acid sequence. Now, I bring that up because structural proteins and their primary structure is unique. And they're unique because they're kind of not unique. They generally have a repetitive pattern, which I suppose that's kind of redundant there, but repetitive pattern. A pattern is by nature repetitive. So what that means is that the primary structure of a structural protein would be something like leucine, arginine, alanine. Leucine, arginine, alanine. This is just an example, just to illustrate, what I'm getting at. And they will have LRA, LRA. So the peptide is those three amino acids, but like I'm gonna put this in parentheses, 600 times. So it's well, those three, or actually those one, B, 9, 12. Those 12 amino acids repeated 600 times over. Okay, well, and that's kind of the nature of structural proteins. They have simple, repetitive sequences. That's what I've got for you guys today. I wanted to say thank you guys very much for coming to class. I also want to say thank you guys for bearing with me as I was coming up with what I think is a pretty clever idea of communicating structural signal transduction. Um, and I'm just thinking of a bar as some place that people don't always get into and, want to communicate messages. Um, so I hope that this made sense. But other than that, that's what I got. I hope that you guys have a great afternoon. I've got office hours starting in about 30 minutes. So I hope you guys have a great weekend. I'm going to go ahead and stop my share, and I'll be stopping my recording in a moment.